Today I am doing a presentation on your UC Davis certificate. The goal of this presentation is to teach you what the UC Davis certificate will tell you just by looking at it. If you have any questions, feel free to email the email address support@betterbred.com. So what can you tell from just looking at your UC Davis certificate? Here it is. This is what you get in email, and it can be pretty daunting on your first glance. So what's at the top? This is the information about your dog. Most of the information is pretty self-explanatory. However, the case and report ID are what you will give us right here and right here at Betterbred so that the data may be uploaded and analyzed on the Betterbred website. And it's fairly simple. All you have to do is make a public membership and give us these numbers and we will upload it and analyze it for you. With that owner's permission, when you enter on Betterbred, you at UC Davis willingly shares the information with us to help us aid breeders. We have a good working relationship with them. Okay, so what's the middle left area with a pretty bell curve? That's this. This area is called internal relatedness, or IR. This region of the certificate tells you where your dog's IR falls in comparison to the rest of the tested population. This particular dog has a very low IR over here, and therefore that dog is outbred. A high IR dog over here would be considered inbred. Notice that the color goes from green to orange-red. This indicates less inbred to more inbred. Now, something that you should know and is very important is an outbred dog could have very common genetics for the breed or unique genetics. IR does not quantify rare or common genetics. So, I've talked about IR, but what is IR? Now, one thing to know is there will be a whole presentation on IR. So if it doesn't make sense to you right away, there's another presentation that you can watch to figure out a bit more about IR. IR stands for internal relatedness, and it's a fairly complex calculation based on allele frequencies and heterozygosity. Essentially, it's a measurement to demonstrate whether an individual dog is inbred or not. Now remember, two dogs may have the exact same IR but have very different genetics. All right, so what's the middle right area with lots of rectangles? This is a dog's STR markers. So what's an STR? STR stands for short tandem repeat. This is a region of DNA that is known to have repeating sections. Population-wide, there's many different numbers of repeats. The different number of repeats are each designated as a specific marker. In this table above, different markers are indicated by the numbers in each square, so a rectangle. So here, this is one marker. These are other markers, and these are different locations on the chromosome. So location 1, 2, 3. So each one of these points are different points in the chromosomes, and each different number here is a different type of variant of that gene or marker. It's very important to note that these are not actually genes that express for anything. They're not meant to do anything, and they were chosen by UC Davis for that reason, because the whole point is to track aspects of a dog, but not to select for any particular traits. So the STRs that were chosen by UC Davis are from the International Society of Animal Genetics. They are markers that are used often to determine paternity. And there's also some proprietary markers that UC Davis has developed on its own. Okay, so this box contains 33 areas of the genome that's being measured. You'll see 1 through 33 listed at the top here. Below each number, 1 through 33, are two boxes. These indicate what alleles and markers are at each location. If the boxes are both white, that means they contain the same STR. This means that this region of DNA in your dog has the exact same genetic material. That's called homozygous. If the boxes are multicolored, they indicate that your dog has different STRs in this region of DNA and therefore has different genetic material. This is called heterozygous. And each one of these genes is received from your sire and dam. We can't tell which one is from which sire or dam. So here's a visual of short tandem repeats. This is one section or one chromosome. This is another chromosome. So this dog has 5 repeats here. or this is one chromosome, this is another chromosome. So on this chromosome, this dog has 5 repeats. On this chromosome, this dog has 6 repeats. So at each location, every breed has anywhere from 2 to 27 different variations of repeats. So this is one variation, this is another. These are STR markers or alleles that are reported, so the numbers that they give. Now, the numbers don't mean that there's 187 different allele markers. It's just an arbitrary number that they've assigned. Again, this dog is heterozygous because it has 2 different types of repeats. If it had the same, it would be homozygous. So homozygous, both markers are the same. Heterozygous, the markers are different. STR markers continued. The STRs and their frequencies are used to calculate IR. What do I mean by STR frequency? Each marker will show up in the population a certain percentage of the time. UC Davis tracks this and provides it in a table right here. So here is the first section right here. And here's the frequency of the alleles that show up in the population for Alaskan Klee-Klays. These frequencies are used in a number of calculations at Better Bread, but on the certificate, it is used to calculate the IR, which was discussed earlier and we will discuss in more depth in a future presentation. Above, you can see that the reported frequencies for the first marker, as we talked about, this dog has 2 copies of 94, which appears in 21.2% of the tested alleles in the population. What is the area below the boxes with the letters DLA? DLA, dog leukocyte antigen. The major histocompatibility complex is the region of DNA that contains the immune system. In dogs, this region is called the DLA, dog leukocyte antigen. There are 3 of these regions, and UC Davis tests 2. Now, I should note that while UC Davis tests 2, this is currently the UC Davis test currently tests the most areas of the immune system of any other test out there. In theory, the more diverse the haplotypes, the healthier the dog. UC Davis recommends selecting for 2 different haplotypes of DNA in your puppies. More intensive research was done with standard poodles in the DLA with disease associations. So let's review. Right here is the IR bell curve and IR value. This tells you how inbred your dog is or outbred. The further to the left, the more outbred. The further to the right, the more inbred. And again, this doesn't tell you how common your dog's genetics are for your breed or uncommon they are for the breed. The STR panel here are the frequencies of alleles that are used to calculate IR and many other measures on the Better Bread website. And below here are your dog's DLA types. Questions? Feel free to email us at support@betterbred.com. And if you have a dog to submit from either the test phase of research in your breed or one that you've done currently, you can submit at betterbred.com/submit. The next presentation will involve more detail regarding IR and its uses.